Welcome back to the deep dive. Today, we are stepping into territory that feels, and I don't use this word lightly, revolutionary. We are talking about the moment where biology stops being something we just study and, starts being a language we can actually speak.
It's a massive shift.
Yeah.
I mean, we are moving from reading the code of life to literally writing it.
Yeah.
And specifically using generative AI to
Yeah.
do the writing.
But let's be clear right off the bat, we aren't talking about ChatGPT writing a poem about DNA. We're talking about a model that hallucinates, for lack of a better word, actual functional genetic code.
Right.
We're covering a paper from Nature Genetics called Designing Synthetic Regulatory Elements Using the Generative AI Framework DNA-Diffusion.
That's the one. And the core promise here is, well, it's wild. They claim they can generate synthetic DNA sequences that are not only novel, meaning they don't exist in nature,
Okay.
but are actually better at controlling genes than the DNA that evolution spent millions of years perfecting.
That is the hook. Better than nature. But before we get to the part where they essentially cure leukemia in a test tube, which we will get there, we need to set the stage. Why do we even need to write DNA? I thought the big thing was the Human Genome Project. We mapped it. We know the letters.
We did map it. We're excellent readers. We know where the genes are, and we've mapped millions of these things called regulatory elements.
So what are those exactly? The switches?
Think of them as the switches, yeah, or dimmer knobs that turn genes on and off.
Yeah.
But here's the bottleneck. We can read the switches, but we can't build new ones from scratch.
Because we don't really understand the grammar.
Exactly. If I want to build a gene therapy that, say, kills a cancer cell but leaves a healthy liver cell alone, I need a very, very specific switch. Right now, I have to go scavenging through the genome, hoping to find a natural one that sort of fits. I can't just type out a new one.
So the solution in this paper is don't even try to learn the rules yourself. Just build an AI, feed it all the data we have, and let it figure out the rules.
Precisely. They built a model called DNA-Diffusion. And if you follow tech at all, you've heard of diffusion models. It's the exact same architecture behind Midjourney or DLLE.
The image generators. You type cat in a spacesuit, it gives you a cat. But this is where I get a little skeptical. An image is pixels. It's colors. You can blend them, make them fuzzy. DNA is discrete. It's A, C, T, G.
Yeah.
It's not fuzzy. How do you apply blurring logic to hard code?
That is the first major technical hurdle they had to solve. To get it, you have to understand how a diffusion model works. So imagine you have a clear photo. You slowly add Gaussian noise, basically static, step by step, until the photo is just gray snow. That's the forward process. The AI's job is to learn how to watch that movie in reverse. It learns to take the static and mathematically scrub it away to reveal the original image.
Right, that makes sense for a photo. But you can't add static to the letter A. It's either an A or it isn't it?
You're exactly right. So they had to translate biology into math. They used a technique called one-hot encoding. Now, usually you'd use zeros and ones, but they did something really clever here. They encoded the nucleotides, the A, C, T, G, AS values ranging from -1 to +1.
Why the negative numbers? What does that do?
It creates a continuous mathematical space.
Yeah.
It lets you introduce that gray area you were talking about. By using a range, you can take a sequence and mathematically blur it. You can make an A look a little bit like a C and a lot like random static.
Ah, okay. So it allows the diffusion model to apply its physics.
It does. It can take pure random noise and denoise it back step by step into a crisp, discrete DNA sequence. They basically trick the AI into thinking DNA is an image.
That's brilliant. So what's the brain of this thing?
The brain is a UNet architecture, pretty standard for this kind of deep learning. It takes 3 inputs. First, the noisy sequence. Second, a time step, which tells it, hey, we're at step 10 of 50, so scrub harder. And third, and this is the most important part, a cell type label.
That's the prompt. Like, make me a liver switch.
That's it. They trained this model on something called the DHS index. These are regions of the genome that are physically open and active, and they focused on 3 extremely different cell lines to prove it could be specific.
What were they?
They used GM12878, which is a B cell, so a blood cell, then HepG2, a liver cancer cell, and K562, a leukemia cell.
So 3 totally different environments. I mean, a liver cell and a blood cell have completely different operating systems.
Right, and that's the point. If the AI works, it should be able to write a sequence that screams, turn on in a liver cell,
Right.
but whispers, stay quiet in a blood cell.
Okay, so they run the model. They start with random noise, give it the liver prompt, and after 50 steps of denoising, out pops a 200 base pair sequence. They made 100,000 of these.
For each cell type.
Right. Now, here is my second skeptical question.
Is it just plagiarizing?
That's the memorization check. It's a great question. Because if I ask an AI to write the opening line of a famous novel about a whale, it's going to say, "Call me Ishmael." It didn't create that, it memorized it. Is DNA diffusion just spitting back chunks of the human genome it saw during training?
They tested this using a BLAT analysis, which basically just aligns the new sequences against the known human genome to look for matches. The results were startling.
How so?
Only 4.7% of the AI-generated sequences had any significant match, and they defined that as more than 20 base pairs overlapping.
4.7%, that seems incredibly low.
It is. For context, if you just threw a dart at the genome and picked random coordinates, you'd get about a 23% match rate just by pure chance. So the AI is actually less likely to copy the genome than random noise would be.
So it's genuinely writing new code. It's composing original music, not remixing old tracks.
Exactly. But that leads to the next problem, right? Just because you wrote a new sentence doesn't mean the sentence makes any sense. The purple elephant danced on the moon is a novel sentence, but it's not a useful biological instruction.
This is where the paper introduces my favorite analogy, the avocado pizza problem. I think this really helps visualize the challenge here.
It's a brilliant heuristic. If you ask an image generator for an avocado pizza, you have two distinct constraints. First, the pizza constraint. It needs to be round, have a crust, maybe some sauce. That's the grammar of a pizza.
If it gives you a square block of cement, it failed.
Right. In biology, this means the DNA sequence must look like a regulatory element.
Yeah.
It has to invite proteins to bind to it. That's the pizza. The second constraint is the avocado. That's the specific flavor. It needs to feature avocado, or in our case, it needs to be specific to the liver.
So you need the general grammar of a switch plus the specific vocabulary of a liver cell.
Exactly. So did the AI learn the vocabulary? To check this, they looked for motifs. Motifs are like the words of DNA. They're short patterns that specific proteins called transcription factors bind to.
And what did they find?
Well, they looked at the liver sequences, the ones for HepG2. There's a very famous protein called HNF4A. It's a pioneer factor. It's basically the master key for turning on liver genes. In natural liver DNA, you find the motif for HNF4A about 29% of the time.
Okay, so that's our baseline.
In the AI-generated sequences for liver cells, it appeared 47% of the time.
Wow. So the AI didn't just learn HNF4A is good. It decided HNF4A is great. Let's use a lot more of it.
It hyper-optimized. It figured out that to be the most liver-like sequence possible, it should pack that motif in. And it did this spontaneously. No one told the AI HNF4A is for liver. It just figured it out for the patterns in the training data.
And it wasn't just keywords, right? I read that it learned sentence structure, too.
Yes, the syntax. For the B cells, it learned that the motif for a protein called IRF1 and another called STAT1 usually appear together. But crucially, it maintained the correct physical distance between them. If they're too close or too far apart in the DNA, the proteins can't interact properly.
And the AI preserved that spacing.
Perfectly. It implies a deep functional understanding. It's like it derived the 3D physics of protein folding just from the statistical probability of letters on a line.
That is mind-blowing. But as we always say, in silico, predictions are cheap. A computer saying this will work is very different from a cell saying this works.
Very different.
So let's do a quick recap before we hit the wet lab. First, the goal is to use diffusion AI to write new DNA switches. Second, the method is to treat DNA like an image blur with noise, then teach the AI to clear it up, but with a specific cell type in mind. And so far, the result is that it creates novel sequences, no plagiarism, and it spontaneously learned the correct grammar and vocabulary.
Perfect summary.
Yeah.
So now the wet lab validation, they used a method called StarSec.
To break that down for us, StarSec.
It stands for Self-Transcribing Active Regulatory Region Sequencing. It's a bit of a mouthful. But think of it like this. You take about 5,800 of your best AI-generated sequences. You synthesize them into real DNA, and you stick them into plasmids. These are little loops of DNA.
Okay. And then you put those loops into the real cells.
Exactly. The setup is designed so that if your synthetic switch turns on, it forces the cell to transcribe the switch itself into RNA. So you basically have a microphone. If the switch works, it screams. You just count the RNA tags.
Yeah.
The louder the scream, the stronger the
verdict. Did they scream?
They worked beautifully. The sequences designed for liver only screamed in liver cells. The sequences for leukemia only worked in leukemia cells.
Yeah.
Specificity was a success. But here's the kicker.
What?
The top-ranked synthetic sequences consistently outperformed the strongest known natural positive controls.
They were louder than nature.
Significantly louder. The AI created enhancers that were more potent than anything we found in the natural genome for those cell types.
Okay, I have to play the skeptic again. StarSec is great, but it's artificial, isn't it? Those plasmids are episomal. They're just floating around in the cell juice. They aren't integrated into the actual hard drive, the genome itself. They aren't dealing with all the chromatin folding, the real mess of life.
You are absolutely right. StarSec is like testing an engine on a stand. It tells you the engine runs. It doesn't tell you if it runs when you bolt it into a car and drive over a pothole. To prove this really works, you need endogenous modulation. You have to put the code into the real genome.
And this leads us to the climax of the paper, the AXIN2 experiment. This is where the rubber meets the road.
This is the real deal. They chose a target gene, AXIN2. And this is a high-stakes choice because AXIN2 is a known leukemia-protective gene. If you can boost this gene in B-cell leukemia, the patient has a better chance of survival.
Is there a natural benchmark here, something to compare it to?
Yes. There's a specific genetic variant found in some humans, a lucky mutation, you could call it. People who have it naturally make more AXIN2, and they tend to survive leukemia longer. That is the gold standard that nature has provided.
So the matchup is the AI versus evolution's best shot.
Exactly. They used a gene editing tool called ExtraAsec to physically splice the AI-generated sequences into the actual AXIN2 location in the genome of leukemia cells. They literally replaced the natural switch with a synthetic one.
And what happened?
First, the specificity held up. The liver-designed sequences did absolutely nothing in these blood cells. That's good. It means no off-target effects.
Right.
But the B-cell designed sequences, they successfully reactivated the gene.
Okay.
And this is the headline. Multiple synthetic sequences drove gene expression higher than the natural protective patient variant.
Wow. That's heavy. We aren't just restoring function here. We are enhancing it.
Yeah.
The AI looked at the problem and designed a solution that's better than the best accident nature ever came up with.
It kind of validates this superbiology hypothesis. We can engineer regulatory elements that are objectively better at their job than the native code.
But wait, did they all work? I imagine biology is messy and it probably pushed back.
It did. Not every sequence worked. And the failures are actually super instructive. They found some sequences that successfully opened the DNA. They cleared the way for transcription, but the gene didn't turn on.
So the door was open, but nobody walked in.
Exactly. Or maybe the wrong people were blocking the door. When they analyzed the failures, they found they often contained binding sites for repressors. These are proteins like RUNX2 or LF1. They're like the bouncers of the cell.
So the AI inadvertently included a stop sign right next to the go sign.
Right. It shows that just having open chromatin isn't enough. You need the perfect cocktail of activators, and you need to rigorously exclude the repressors. The AI is still learning that nuance.
That's a fascinating detail. It shows the complexity. It's not magic, it's probability. So let's zoom out a bit. DNA diffusion isn't the only game in town. How does this compare to other attempts, like GANs?
They did a comparison to Jan's generative adversarial networks and another tool called CODA. They visualized the results on a radar plot, which is like a triangle with three corners: diversity, activity, and specificity.
And who won that fight?
Well, CODA turned out to be a one-trick pony. It had high activity, but almost zero diversity. It basically found one sequence that worked really well and just kept repeating slight variations of it.
Oh. And the JANs.
The JANs had high diversity, lots of creative ideas, but the activity was low. They were imaginative, but weak. DNA diffusion was the only one that really stretched the triangle. It offered high diversity and high activity, a good balance.
And there's a feature mentioned in the paper, some kind of knob you can turn to control this.
Yes, the CFG scale, classifier-free guidance. This is a game changer for design. If you set the scale to 0, you're telling the AI to be natural. It mimics the training data. It writes DNA that looks like it belongs in a human.
But if you crank the volume up...
If you turn it up to, say, scale 7, you're telling the AI, don't be natural, be optimized. You force it to focus purely on the features that drive the highest probability of success. At scale 7, the sequences stop looking like normal biology and start looking like supercharged machines.
So you're sacrificing naturalness for raw power.
Exactly. You get sequences that are less diverse but incredibly potent. It becomes less of a generator and more of a sequence optimizer.
Okay, so let's bring this all home. We've gone from theory to avocado pizzas to, well, essentially treating leukemia in a dish. Why does this matter for the listener who's, driving to work right now?
It matters because of the delivery problem. Right now, a lot of gene therapy uses viruses, like AAVs, to deliver cures. Think of AAVs as a delivery truck. But it's a tiny truck. It has very limited cargo space.
And natural switches are big and clunky.
They can be thousands of base pairs long. They take up half the truck. These synthetic elements, they're only 200 base pairs. They're compact and potent.
So you can fit more medicine in the truck.
Exactly that. And because they're so specific, you could inject a therapy into the bloodstream that travels everywhere, heart, lungs, brain, but it only turns on in the tumor cells. You minimize side effects not by where you inject it, but by how you code it.
That is the holy grail of precision medicine. But it leaves me with a sort of a philosophical itch. We are crossing a pretty major line here, aren't we? I mean, the AXIN2 result proved we can outperform the lucky natural variant.
It does raise a profound question. Traditional medicine is restorative. You break your leg, we fix it. You have a mutation, we correct it back to the human baseline. This technology suggests we can engineer states that are better than the baseline.
We aren't just fixing the code, we're upgrading it.
We're entering an era of optimization. If we can write code that fights cancer better than millions of years of evolution could come up with, where does that stop? And do we optimize metabolic genes to prevent obesity? Do we optimize muscle growth?
And the real question for you listening is, would you trust a piece of code written by a hallucinating AI to run your own operating system?
It's a question we might have to answer sooner than we think.
A fascinating, maybe even a little frightening glimpse into the future of synthetic biology. The paper is Designing Synthetic Regulatory Elements Using the Generative AI Framework DNA-Diffusion. If you want to see that radar plot or the avocado pizza diagram, the link is in the show notes.
It's definitely worth a read.
Thanks for diving deep with us. We'll see you on the next one.